Hi, everybody. I'm Dr. Austin Perlmutter. I'm the Chief Science Officer here at Big Bold Health. And today I have the privilege to get to speak with all of you about a really interesting topic, and that is the intersection of longevity science and the microbiome. Now, if you've attended other webinars that I have been putting on, or if you've been learning about the science of longevity, you've probably heard a lot about the various pathways linked between longevity, diet, and other aspects of lifestyle. And today we're going to be getting into the most recent information specific to the connection between the microbiome and longevity. And we'll talk about this at length as it relates to why longevity science may be getting certain things wrong in terms of the public conversation, where we can go for better explanations, and why the microbiome is such a critical part of the conversation as it relates to what we can do to slow our rate of aging, and in particular to enhance health span. So not just years of life, but the quality of those years. Quick disclaimer here. This content is just for educational purposes. I am not your treating physician. This is not designed to take the place of medical care. You should follow up with your healthcare practitioner before making any changes to your healthcare protocols. Again, this is just for educational purposes only. So what are we going to talk about today? I'm going to be a little bit ambitious in this presentation. We're going to cover a lot of material. And so if there are things, hopefully, that you haven't heard about before where you say, hey, that science is a little bit too complicated, or I'd like some more time with that. We will be sending this out afterwards in terms of if you're watching live, and if you're watching this after the fact, obviously you have plenty of time to go through and look at all of the research that I'll be citing here. But just know that the goal here is not for you to understand everything about longevity and the microbiome, but rather to get a good sense as to the basics of the research around aging and the microbiome, and then to get a sense as to what the most recent research is telling us so that you can be prepared for what the future looks like as it relates to the connection between the gut and longevity, and specifically the microbiome and longevity. But what we will be talking about today is the connection between longevity and the microbiome. We're going to focus on the most important publications on the topic of gut and microbiome health and longevity, and then we're going to focus on research that has just been published in the last year specific to this topic. We'll talk at the end a little bit about what this represents for the future of science, and then we'll go into some of the practical tools in terms of how does this map onto lifestyle modification. So I wanted to kick off the actual conversation today with a brief review of what we know about longevity science, and in particular what longevity science is telling us in 2025, which I'm sure will be different from what it's telling us in 2028 and is absolutely different from what it was telling us 10 years ago. So key to this conversation and why we're talking about slow aging instead of just longevity is that aging doesn't occur at the same rate in each person. You can have two people who have lived the same number of years, one of whom is going to live for another 30 years and one who is going to live for 5 more years. And the difference here is in part that our bodies respond differently to those years that we are alive. And so aging doesn't occur at the same rate in each person, nor does it occur at the same rate in individual systems within our body, in particular the immune system, which we'll get into. The number of years you have in birthdays is different from the number of years that your organs experience or your cellular age. And a large body of research, some of which we've actually published on in Big Bold Health, shows that it isn't about how many birthdays you've had. Now, not to take away from the value of a birthday. Birthdays are fun, don't get me wrong, but it's more about how your body weathers those years. And so we can start to understand what are the key pathways that are driving our cells and our organs towards more rapid aging or towards slower aging, so that we can modulate or influence that process and slow the rate of aging. Many of the key ideas around longevity have kind of stemmed from this concept of our genes kind of determining how long we live. And we now know from the research that the majority of what determines how long we live is not genetic, but it is a function of our lifestyle, that and our environment. And so that means that if your goal is to live a long and healthy life, and I hope that's your goal, that's my goal, that the majority of the variables that will determine how long and how well you live are not your genes. For the average person, it is your environment. So we'll be talking about how that relates, in this case, to the microbiome. One of the themes that I bring up in these conversations is that many of the ideas that we hear about in the longevity health space are sensationalized or otherwise incorrect. Longevity science is different from what you will see on TikTok or Instagram or even on the news, because these are always chasing the latest breakthrough, the miracle cure, the fountain of youth. There's tons of amazing science, and it is incredibly relevant to how we should be living our lives, but it isn't this one-shot solution where there's some pill or some other thing that's going to cause us to live to 160 or 180. The reality is that the most important things that we can do to promote a long and healthy life are going to be compounding steps that influence our biology to basically benefit our slower speed of aging each day. And so today we're going to be talking about, in particular, the microbiome. But just to reiterate this point, lifestyle and environment are the most important determinants of how long you live and how well you live. And so that's why there's so much amazing opportunity for us to leverage the science for our benefit. So what is actually driving aging? Now, for those of you who read books like Outlive or Lifespan or follow popular podcasts that are focused on longevity research, you'll know that there are these so-called hallmarks of aging. And these are various systems, pathways within our cells, within our bodies, that have been postulated to drive the aging process. Now, there are new hallmarks each day. Some say there's 12, some say there's 14, some say there are more. But these are some of the most important and most clearly identified hallmarks, drivers of the aging process. And they relate to things like a shrinkage of telomeres. These are these protective end caps on our DNA. Senescence, which is the aging of our cells, chronic inflammation, issues with something called autophagy, which is a cellular cleanup pathway. There are many of these hallmarks of aging. For the purposes of what we focus on in Big Bold Health, we are mostly focused on 6 of these hallmarks. So epigenetics, which is the way that our DNA is used because of marks on top of our DNA. Nutrient signaling, how do the nutrients that we consume influence the aging process? Mitochondrial issues, mitochondria being considered the powerhouses of the cell. They do a whole lot more than that, but changes in our mitochondria are driven in large part by the nutrients that we consume, so you can see how these things connect. Cellular senescence, which is the aging of our cells. Chronic inflammation, which most of you are probably very familiar with, the idea that as we age, we actually develop more inflammation, and this can promote and even speed the rate of aging. And then alterations in the microbiome. So for the sake of making this lecture 1 hour as opposed to 18 hours, we are going to focus today on the microbiome, and we're going to talk about the basics of the microbiome. So many of you may already know some of this stuff, but I think it's really important to set the framework for what we're actually talking about. I will be getting into some terminology. You don't have to remember it all, but just so that you have it available. But we're going to focus in particular on the microbiome as it relates to our rate of aging. So what is the microbiome? We have trillions of microbes that live on and in us, and you can find various studies, but it's somewhere around 38 trillion or so microbes that we believe live on us. And of those 38 trillion, that's actually talking primarily about bacteria. There's probably 10 times more various forms of viruses, but really most people focus on the bacterial component of the microbiome. It is important to note that, like any aspect of science, research, and general focus, we tend to narrow in on what's most interesting and most available, but that's not necessarily the entire story. So by and large, the focus on the microbiome, when you hear about people discussing the gut microbiome or products that improve the microbiome, has been on the bacteria in our large intestine. But we know that there are a number of other forms of microbes. So viruses, archaea, which we'll talk about in a moment, fungi, all of these things live on and in us and influence our health. It's also important to note that we have more species of bacteria that live on and in us than we have different cell types. And maybe even more interesting is that we have about 100 times more microbial genes in our microbiome than we have our own genes. As this relates to more tangible science, we know that the makeup of our microbiome, and so again, usually the focus here is on the microbes, specifically bacteria in our large intestine, but we're now learning about the microbiome on our skin, in our lungs, and other parts of our body, that it relates to longevity and it relates to aspects of both how long we live and the quality of those years. We also know that the microbiome influences our metabolism. So the way when we consume food, that food gets converted into nutrients that we can utilize is in part directed by the microbiome. We actually know that the amount of calories that we may extract from our food in part is a function of the microbiome. And as it relates specifically to the research we're doing at Big Bold Health, we know the microbiome and the immune system, as we'll discuss in a moment, are incredibly closely linked. So just to give you a teaser, I think, of where the future of this is going, we've largely focused on the microbiome in our intestines, but we know that we have microbes living on our skin, living in our nose, living in our mouth, living in our lungs, living even in our brains and other parts of our body. And the research is really just emerging as it relates to the role of those various microbiomes in terms of addressing our health. So here's just a quick prediction from me. The skin microbiome is going to turn out to be incredibly important to the quality of our skin. So things like wrinkles, sure, but also just the relative degree to which our skin is capable of fending off environmental threats, the degree to which it's able to retain moisture. And like many things, we've kind of adopted a system where we use harsh chemicals and soaps to clean our skin each day. And I'm not talking about your hands. I'm talking about facial cosmetics, things we put on our skin. I think we will absolutely recognize the role that the microbiome of the skin plays, and we'll probably have to dramatically pare back on some of these more abrasive or harsh chemicals that have been creating problems with our skin microbiome. All of this is to say the microbiome is located throughout the body. Narrowing in on the gut microbiome, which is again where the majority of the research that we'll be talking about today is focused, and where the majority of the research in general and people's discussion of the microbiome focuses, most bacteria in the body are located in your large intestine. It is the best studied microbiome in the body. And just like when we talk about longevity primarily being an environmental function and not a genetic function, we now know that the makeup of the microbiome is predominantly driven by environment and not genetics. We also know that diet by itself is thought to explain about 20% of the differences in the gut microbiome. So that's about double the contribution that we get from our genetics, which means that as it relates to the makeup of your microbiome right now, diet plays a larger role than your genes, and especially over the lifespan, because we know that obviously our food changes depending on many variables in our lives, but our genes tend not to change. So how does your microbiome influence your physiology? Well, when you eat food, it changes the microbes in your gut. When you eat food, it also influences the immune system, but when you eat food that changes the microbiome, that can then impact the immune system, especially in the gut. The gut microbiome also produces a host of metabolites. We'll talk about this in terms of a definition in just a moment, but realize it's not just about the bacteria in your gut, it's about what they produce that may have an even bigger effect on our physiology. So here's kind of the list of the key terms to know about as it relates to the microbiome. You've heard of many of these before, you maybe have seen them in the store, but we're going to go through these so that you have a sense as to the key nomenclature or the key terminology that you need to know about your gut microbiome. Prebiotics are technically non-digestible fibers that feed beneficial gut bacteria. I would say we need to update that definition a little bit because we now know that molecules like polyphenols can actually act to influence beneficial gut bacteria, and polyphenols are not exactly fiber. Probiotics. So probiotics are the products that you see in the store that talk about the live bacteria or just bacteria in general. Probiotics are beneficial bacteria that when we consume are supposed to improve our gut health. Postbiotics are compounds that are made by probiotics when these probiotics are just undergoing their typical day-to-day functions. So postbiotics are things that probiotics create or Postbiotics are things made by microbes. And then we have synbiotics, which is a newer term. These are products that contain pre and probiotics. And the idea is that when you combine a pre and a probiotic, you may get a bigger effect than if you were just consuming them both in isolation. Then we have dysbiosis, a term that basically means your gut microbiome in particular is usually where it's used, but it can refer to any microbiome, has an unhealthy makeup. And then finally, fermentation. So many people are familiar with fermentation because maybe they do some fermentation in their home. Maybe they purchase products that are fermented. So often discussed in the context of alcohol or perhaps any fermented foods that you might buy in the grocery store. But the basic idea here is that when you give microbes carbohydrates, they can actually break down the carbohydrates and produce various products. So anytime that microbes in the gut or otherwise are eating carbohydrates, we would call that process fermentation. As it relates to what is in your gut microbiome, this is complicated stuff, and you can get lost in terminology. We're not going to over-focus on you knowing the names of a whole bunch of various bacterial species, but it's important to know that generally speaking, we look at phyla. So that's kind of a taxonomy of the various microbes in our gut. It's a layer of taxonomy. And when we look at phyla, there are really two major phyla that kind of constitute the majority of the bugs in your gut. And each of these phyla has within them many different types of species, but Firmicutes and Bacteroidetes are the two dominant phyla in your gut. If you're interested to learn more about all the different microbes in your gut, there's tons of resources that you can get, but it does become quite complicated quite quickly. And in order to make this conversation flow and also to provide you with something of value, we're not going to go into all the various subspecies of bacteria at this time. I do want to dive in, however, to postbiotics. This is really where an emerging subset of research is showing there's an incredible value. So it isn't just that you have a makeup of various microbes in the gut, that you have lactobacillus or Akkermansia. It actually has a lot to do with what those bugs are doing. So if you think about this, it's one thing to have some really great people on your team at work, but it's also important that those great people are producing good work. And so the postbiotics, or the products created by bacteria, are now being studied to have a number of different roles in our health. The most important of the various postbiotics to know about are going to be the short-chain fatty acids. And so short-chain fatty acids are produced by microbes, certain subsets of microbes, when they digest fiber. The short-chain fatty acids that you should probably know about, the most important would be butyrate, but you also have acetate and propionate. Butyrate is important because it's now being studied to have a number of roles throughout the body, on epigenetics, on immunity, on metabolism. It's also a primary fuel source for our gut cells. So when our microbes consume fiber, they make these short-chain fatty acids, and these short-chain fatty acids, we're now learning, may be as important as the actual microbes themselves as it relates to the long-term effects on our health. For completeness sake, I will say that bacteria in our gut and throughout our body can also produce a number of other metabolites. Those include indoles. These are basically metabolites of tryptophan. So for example, we know that gut microbes can produce serotonin. There's a common misconception that the serotonin in your gut has a role to play directly in the serotonin stores in your brain. So serotonin in the gut does not go through the blood-brain barrier, but it can influence pathways like the vagus nerve, and in doing so may influence mental state. But realize that the gut microbes produce a number of molecules. Tryptophan metabolites are one. Bile acids are another set of metabolites they produce that we know influences the immune system. And then trimethylamine is an interesting one because it can convert to TMAO, which some people may be aware of. There's some concern that this molecule can be linked to cardiovascular issues, and some people will use this as reason to promote a more plant-based diet. And some people will also use this to promote consumption of less of a molecule called choline, because choline can actually convert into this TMAO. If we have time at the end, I'm happy to get into that, or if people have questions or comments on that question. put it into the comments and we'll try to get back to that because I know that is a topic of interest to a number of people, specifically those who are curious about plant-based versus animal-based diets. Another system within the gut that is important to know about is the enteric nervous system. So you may have heard this term, the second brain. The enteric nervous system is a branch of the autonomic nervous system that is embedded within the gut wall. It contains over 100 million neurons, which makes it the largest clustering of neurons outside of your central nervous system. And the enteric nervous system picks up data from the gut, sends it to the brain, and also importantly receives signals from the microbiome. I'll also mention kind of just the last background framework here, the vagus nerve. I'm sure you've all heard of the vagus nerve. It's cranial nerve number 10. It is the main physical link between your gut and the brain. It runs in both directions, but the majority of the fibers run from the gut to the brain, where they carry sensory data, and that sensory data is certainly influenced by your microbiome. So important here to say, as we're considering how does the microbiome influence the rest of the body, there are a number of pathways, but one that connects in particular your brain function with your gut function and can help to explain some of the links between brain longevity and gut state is going to be the vagus nerve. Now, I mentioned that we're going to get a little bit technical today. These are some terms that you probably don't need to know for general conversation around the microbiome, but if you're curious and you would like to have a better understanding of the research that we'll be discussing, this is important stuff to know. As we look at how to actually explore the microbiome and the tests that we can do to find out what microbes, what bacteria in particular live in our gut, 16S RNA is kind of the go-to way of determining what is found in the gut. So this is a marker that is found in bacteria that allows for researchers to identify the microbes in our gut or in other parts of our body. There are two additional tests that are becoming more advanced tests, including shotgun metagenomics. So this looks at DNA to identify subtypes of microbes and also gives us a bit of a sense as to the function of those microbes. And then there's metatranscriptomics. So if metagenomics looks at DNA, transcriptomics looks at RNA or transcripts, and it gives us a sense not only of what's happening within the microbiome, but what is actually what are the functions of the microbes, because it's telling us what are they producing. So these are kind of the core ways that we can interrogate or understand the gut microbiome. Now, as I mentioned before, but I'll just remind you, the gut microbiome not only influences other longevity pathways, but it's influenced by other longevity pathways. So all of these, including inflammation, including epigenetics, including alterations in mitochondria, are being influenced by and influenced the microbiome. So there's an interconnected web here. You can't parse out and say, well, you have the microbiome. If you get that into good shape, that's just an isolated part of the system. Each of these things do contribute to overall longevity and health span. So here's an interesting question, which is, how important is the microbiome in general? What if we just had a drug or some other way to completely remove the microbiome? What would happen to our health? And why this is interesting is I, like probably many of you, grew up in a time where we saw these posters of germs telling us about how bad bugs were. We're taught to wash our hands, which is a good thing to do, but we are taught in this kind of context of bacteria are bad. We should get rid of bacteria. So what if we were to just get rid of all the bacteria in our bodies? How would that go? And the model we have here, this is actually a photorealistic image, as you can tell, but this is a germ-free mouse. And so researchers will study the impact of microbes on health by looking at these germ-free animals. Germ-free basically means they are animals raised in sterile conditions. They don't have a microbiome, and they're really only used for experimental purposes. Often these are mice, for all the reasons why mice are typically used. But here's the question. If you are a germ-free mouse, how long do you live? Do you live longer or shorter amount of time than a mouse that is living out in the wild or a mouse that has a typical microbiome in a lab? And the answer might surprise you. A germ-free mouse will live longer than a mouse that has access to or is exposed to microbes in their environment. So does that mean that we actually should try to remove the microbiome? Well, Not exactly. And I bring this up and highlight this as an example of where animal research can't necessarily translate directly into human research if you go one to one. We need to have this extra layer of understanding because we, almost everyone probably who's watching this, live in the real world where we are going to be exposed to environmental threats, pathogens, and other things. And so, in order for us to have resilience, in order for us to function in the real world, we actually need a healthy microbiome. A healthy microbiome is key to regulation of our immune system, regulation of metabolism, regulation of longevity, promoting molecules, promotion of a healthy brain, and actually regulates gene expression. It changes epigenetics. So, point here being, animal research can only take us so far. It can provide amazing insights, but we have pretty much seen consistently across the literature that for real humans in the real world, we actually do need a microbiome, and we need to find ways to make sure that microbiome is as healthy as possible. The majority of what programs the microbiome is environmental. And that means that all of these different things that we engage in each day have a role to play in the setup of the microbiome. That includes our diet, it includes over-the-counter medications, prescription medications, it includes air pollution, birth history. Birth history turns out to be incredibly important in setting up the early life microbiome based on exposure to microbes or not. And then things like alcohol, smoking, and even whether or not we spend time in nature all influence the microbiome. At the end of this lecture, we'll get into the specifics of what that means and maybe some tools for how to leverage this. But to realize that we are each day programming the microbiome with the choices that we make. Undergirding the entire basis of this conversation is that if we understand the microbiome and if we understand how the microbiome influences longevity pathways, we can help to work on slower aging pathways. But one way in which the microbiome has a significant overlap with another pathway related to longevity concerns the immune system. So depending on the study you look at, it does seem like at least 50%, but maybe upwards of 70% of the immune system is located in and around the gut. We also know that the gut immune system interfaces directly with the gut microbiome. And so the way that our microbiome is set up has a direct role to play on the immune system and vice versa. And we know that the gut microbiome can also influence systemic immunity, which is a key driver of how long we age and may be the best way to determine our overall chances at a long and healthy life. So key for this understanding here is to know that the microbiome programs the immune system and vice versa, and this has a role to play in how long we live and the quality of those years. Now let's dig in for just a moment on this microbiome to systemic immunity pathway. So how does what happens in your gut influence the immune system throughout your body? Well, one pathway relates to these postbiotics I've described, these short-chain fatty acids. So when your gut microbiome produces short-chain fatty acids like butyrate, they get into our bloodstream where they can influence the immune system throughout the body. Obviously, a lot of the immune system is located in the bloodstream, but not all. We also know that the microbiome can produce neurotransmitters and hormones. These can bind to and influence immune cells. In addition, because the microbiome has such a major role to play in how we metabolize or use food, and we know that the way that metabolites from our food get into our bloodstream and influence our physiology also applies to the immune system, this is another important pathway to be aware of. And then the microbiome can actually program the immune system in specific organs. And the one that I'm most interested in is the brain, because we know that the brain has roughly 20 billion immune cells called microglial cells, and these cells are responsive to the microbiome. So just to put that together more concretely, the microbes in your gut influence the immune cells in your brain, and the immune cells in your brain influence things like mood, cognition, risk for conditions like dementia. So what you see here is that what happens in your gut microbiome is a major variable in relating to your systemic immunity, and in doing so is a major variable in relating to multiple aspects of health and longevity. Okay, so that's all the preamble. That's all the background information to get you ready for the most recent research in how the microbiome relates to slow aging. So with all of that said, I'll just take a quick moment here, which is to say, if you are not subscribed to this channel, consider subscribing to this channel. So that's for people watching this later. And if you have questions or comments, leave them here. We'll try to get to them. And just thanks a lot, everybody, for caring about this topic. Okay. So let's jump into the research. Let's talk about what do we know? What are we learning about the microbiome specific to aging? Now, the research really got started back in 2016 when this publication came out. So this was in a Nature journal called Genome Medicine. And they looked at the correlations between the gut microbiome and frailty, so in older people. They looked at 728 female twins, and what they did was using this, again, 16S ribosomal RNA, which is how you determine what microbes are in the gut. They were able to make correlations between frailty and gut microbes. And what they found is more frailty correlated with a lower diversity of the gut microbiome. So let's make that more clear. The gut microbiome has lots of different microbes in it, but general diversity of the gut microbiome is seen as a good thing. In this case, what they found is that frailty was related to diversity in that people with less gut microbiome diversity had a higher risk of frailty. Frailty is an incredibly important marker because obviously it correlates with longevity and aging. When we become more frail, we're at much higher risk for dying early, as well as a host of other health-related concerns. So frailty is really a surrogate here for health span. So this was one of the first signals that the microbiome related to a longevity-specific pathway. Now let's fast forward here 5 years to a study that was published in another Nature journal. This is Nature Metabolism. This is 2021. And what they found here is that there were correlations between the gut microbiome, healthy aging, and survival. So let's see what they found. This was an exploration of the gut microbiome in over 9,000 people. They had a good variety of ages of adults from 18 to 101 years of age. What they found is that the people who were healthiest over the age of 80 had consistent changes in their gut microbiome. And what they had is lower Bacteroides composition. Now, we're going to talk about this as an example of why it's complicated to try to understand specific types of microbes in the gut. So we'll come back to this. But they also found in this study that the changes in the gut microbiome correlated with changes in the blood. So what does this tell us? It tells us that people who were healthier agers had specific differences in their gut microbiome compared to people who were not healthy agers. They found that those related to a specific type of microbe, in this case Bacteroides, and they found that the changes in the microbiome correlated with changes in the blood, meaning that they could see that those alterations in the microbiome seemed to translate into pathways within their bloodstream, helping us to understand that what happens in the gut doesn't stay in the gut. So fast forwarding here another year, this is a review paper. So for anybody who's interested to get a general sense as to the research as it was published in 2022, it's another Nature journal, Nature Reviews Gastroenterology and Hepatology, aka GI and liver health. What they discussed here is that the gut microbiome transduces environmental signals and then modifies our risk for disease across ages, and it also changes with our age. Let me break that down a little bit. The gut microbiome responds to what we do in the world. So the food we eat, the exercise we get, the gut microbiome is listening. It changes its makeup. That change in makeup changes our risk for developing health issues. And that change in makeup alters over the course of our lifespan. So what they said here in this quote is resetting gut microbiome derived signals of unhealthy aging through personalized or subpopulation level microbiome associated interventions is a new area of research. And what that's starting to discuss here is if the microbiome directs our rate of aging, if it directs our rate of health conditions, and we can change the microbiome, if we can push it towards a healthier state, then by doing so, we may be able to slow the aging process. And I want to be clear on a point here. So this was published in 2022. We're going to get into a bunch of new research that's been published since then. Mechanistically, all of this makes sense. If we say the gut microbiome is driving at least some part of the aging process, then changing the microbiome may be able to mitigate some aspect of premature death and poor longevity. That doesn't necessarily mean we know exactly what to do. If you just take this microbiome boosting probiotic, you're going to live a longer life. But this is the start of that understanding. What we'll talk about later in this is how do we influence the microbiome for the better to promote this slow aging pathway. But I will just be cautious in saying the takeaway from this lecture is not that we have solved for a single bacterium, that if you just eat a bunch of it, you're going to live forever. Rather, what we're saying is the microbiome is a key part of understanding longevity, and we're starting to get insight into how we may be able to modify the microbiome to actually slow the rate of aging. So this next study, this is a study published in 2023. This is looking at longevity of centenarians, so people who live to age 100 or longer, and how that relates to people's microbiome. So this is a study where they had 1,575 people, of which around 300 were centenarians. So they were looking for, in particular, people who had survived to a very late age. And what they found is that people had, when they were centenarians, when they made it to age 100, they had more youthful microbiomes, and they had more Bacteroides and higher levels of healthy bacteria, lower levels of harmful bacteria. This may trigger in you, as it did in me, a little bit of a challenge, because what we're saying here is that the healthy older people had more Bacteroides. And what I said in the last or 2 slides ago was that the older healthy people over age 80 had lower Bacteroides. So what does that mean? Well, let's get into this for a second. This is a great representation of where the complexity of the microbiome can create confusion. And so what is Bacteroides? Well, it is a grouping of bacteria in the gut. There are over 30 species. Bacteroides can do great stuff for us. It can help to modulate the immune system. It can produce short-chain fatty acids, as I mentioned, those postbiotics that seem to be helpful. It can also metabolize bile acids, but it can also produce molecules like lipopolysaccharide, or LPS, that may be harmful to the immune system. Various ways of looking at this. So when you look at a larger level, Bacteroides tend to be higher in younger people. But some of the research here would indicate that what these species do is they consume the mucus layer of the gut. Now, I know we're getting technical, just bear with me, this is an important example. So Bacteroides can consume the mucus layer, and older people have less gut mucus. So as we look at healthy agers and what they have in their gut, is it possible that really what it's telling us is more about, your mucus layer and whether or not you have the healthy mucus to sustain a population of Bacteroides? Or is it that Bacteroides is actually a reflection of other things happening with aging, meaning that a person's degree of aging, healthy or not, isn't because of the Bacteroides, but Bacteroides is a function of other things happening in the aging process. The bottom line and the point I'm trying to make here is it's tempting to try to pin it down and say, well, this study found this change in bacteria, therefore that's the goal, is to boost it or to deplete that bacteria. But the reality is we're starting to get a whole lot more data on how these changes relate to various pathways within the body. And that's why looking not just at the bacterial population, but when we look at metatranscriptomics or metagenomics in terms of what is actually being produced by, what are the pathways being acted on by the bacteria, that may be even more helpful. So that's to say, we're starting to see some consistent changes, but there's still plenty of science to still get through. So let's jump into some more recent research looking at how correlations between the microbiome relate to aging pathways. So here's a publication from Cell Press. This is 2024, and it was looking at how if we take big data sets, big data from the gut microbiome, that we may be able to use the microbiome to predict a person's age. Now that's really interesting. So many age clocks have been produced over the years. You maybe heard me talk about some of the epigenetic age clocks that tell us about not just our age and birthdays, but rather our cellular aging or organ-specific aging. So what they did in this study is they pooled data from over 90,000 people, so a big data set, and they created a clock that was able to basically look at a person's microbiome and then tell the researcher, here's how old we think this person is, how many years and birthdays they have. They had, similar to the other study, a huge range of populations, but unlike the other study, they were able to capture very young people, so less than 1 year of age to up to 104 years of age from over 60 countries. What they did not find is that a person's diversity of microbiome or richness of microbes predicted their age. But what they did find is that you could predict a person's age based on their microbiome. Now, it's not the most consistent age clock. There are better age clocks if that was the goal, but it's the first time that we've seen that looking at a person's microbes, you can predict a person's age. One interesting correlate from this study is they found that those who were eating a more vegetarian diet would be predicted to have a lower age, and those eating a Paleolithic diet, aka a paleo diet, would be predicted to have a higher age. What do we take from that? What does that mean? It's still a little bit early to make sense of exactly the implications for what that would mean in terms of dietary recommendations. But I would say, as we look at microbial diversity in a healthy microbiome, if we talk about what drives healthy microbes and we talk about these prebiotics, these are things that typically come from plant-based foods. And I think it's pretty consistent that despite certain subpopulations perhaps benefiting from lower fiber intake, that most people benefit from a diet rich in plants and rich in dietary fiber, which supports a healthy microbiome. So moving along here, now we're getting to research that has been published in 2025. So that's this year, if you're listening this year. This is a study that's looking at something called Mendelian randomization. And it's looking at Mendelian randomization to help to get a sense as to directionality and potentially even causality of the link between aging and the microbiome. So here is Gregor Mendel. You may remember him from any genetics course you took or even some basic biology courses that you took. He did a lot of the initial work understanding how genetic traits are passed down. Mendelian randomization, however, is a very new form of science. And what it does is it looks at changes in gene variants that correlate with various outcomes. That's complicated. Let me break it down. What we're trying to always determine is, are two things linked because they just happen to happen at the same time? So this is looking at things like confounding variables, or are they linked because something actually causes something else? So a popular one would be to say coffee drinkers, let's say they were at higher risk of having lung cancer. So we could say that it's because people who drink coffee are, something about the coffee causes lung cancer, but there may be a confounding variable. In this case, there's let's say that coffee drinkers are more likely to smoke. And so smoking is what causes the lung cancer. We don't want to blame coffee consumption. And I certainly don't, because I love my coffee. Point here being, we want to be able to determine what is actually driving an outcome. And Mendelian randomization basically looks at a person's DNA, and it says, based on that DNA, we can predict that a person will have, let's say, a high level of HDL, high density lipoprotein, or the healthy cholesterol levels. And so if we know based on their genes that they are going to have a higher HDL level, then if we look at their heart outcomes and we can see that their heart outcomes are better or worse, we can draw directly from this correlation, which is genetically predicted HDL predicts the heart health outcome. And we can start to determine whether this is causal, meaning it's actually causing the heart health outcome, or if it's not all that related. So all that is to say, in this study, they were trying to determine the potential causal factor here, which was, does the gut microbiome relate to aging? And what they found here is that there were gene variants, so alterations in the DNA that related to different states of microbiome, meaning that DNA in this case seemed to drive certain microbial patterns. They had to find this. This was the only way the study was going to work. As I've said before, and I'll reiterate, the majority of your microbiome makeup is not driven by DNA, not driven by your genes, but there are certain aspects that are. So here they looked at the makeup of the gut microbiome, they looked at frailty, and they looked at these epigenetic age clocks. And what they found is that the makeup of the microbiome related to age acceleration using these epigenetic age clocks. I apologize, I know this is technical, I will do an explanation in just a moment. What they found additionally is that there were links between the makeup of the microbiome and risk for frailty. So what all of this means is they were able to show here that the makeup of the gut microbiome may actually drive aging and frailty, and not just be, oh, well, incidentally, people who are older have changes in their microbiome. So this is some of the first data to suggest that directionally speaking, the microbiome may actually force people towards higher risk of healthy or unhealthy aging. Really important stuff. Again, I know it's a little bit convoluted, but that's really what we're saying here, which is that we may see a directional and potentially causal pathway, not to say that your rate of aging or your age in general is directly caused by your microbiome only, but that the microbiome is actually driving some percentage of a person's aging rate and their frailty. I wanted to expand a little bit beyond the gut microbiome in the way that it's typically described in terms of microbes and various microbiomes in the body. And so a good example of a study that just came out this year was looking at archaea and looking at how archaea can actually help us to understand the correlations between the microbiome and aging. So archaea are single-celled microbes. They're often confused with bacteria and actually kind of only recently have been subsetted out as a different group of microbes. What's interesting about archaea is they are extremophiles, meaning they can exist in extreme environments. And in our GI tract, the majority of archaea are what are called methanogens because they produce methane. So in this study, where they were looking at archaea and correlations with aging in the gut, they did about 250 people, and they looked at their metagenomic data. So again, this is DNA that is pulled from the stool sample. And what they found here is that archaea in centenarians, people who lived to 100, looked like younger people's samples of archaea. They found higher amounts of a specific type of archaea, and they found also that centenarians, people who lived to 100 or longer, had higher production of genes related to butyrate production. Let me unpack that a little bit. We saw earlier some data suggesting that the microbiome of people who survive to a long-lived or basically who live longer correlated with the microbiome of people who were younger, suggesting that one of the ways that people may be protected against aging is a more youthful microbiome, but that was for bacteria. So this is the first time we're seeing something similar for another subset of microbes, in this case, archaea. And what's interesting about this is they also found that one of the pathways may be a production of more butyrate, because archaea, interestingly enough, is also linked to short-chain fatty acid production. Now, if we expand this a little bit outside the large intestine, which again is the place where the majority of our microbes live, here's a study that just came out in July of 2025. So I think technically we're still in June. So this is actually looking ahead to a publication that is as current as it gets. This study looked at differences in the oral microbiome. So in our saliva, you can actually look for various microbes. And they looked between elderly people and adults. This is a systematic review. They used the 16S RNA sequencing again. And what they found here is some inconsistency in certain changes, but a consistent decline in certain bacteria and a consistent increase in elderly people in their bacteria. So this is some of the first data suggesting that as we age, it's not just our gut microbiome, meaning the large intestine microbiome that changes, but rather the oral microbiome that changes. So there's all sorts of things we can understand in terms of where the future is going. But I want to kind of highlight as it relates to some actionables or some take-home messages. What do we do about this right now in terms of our understanding? We know that the microbiome is predominantly being influenced by environmental variables, the things we do, the choices we make, what we're being exposed to each day. So let's talk about, in the last little bit of this presentation, what we know. So as it relates to diet, there's a whole lot of research out there. There are a whole lot of opinions out there. But what we know is that, generally speaking, a diet that is rich in plant-based foods, high-fiber foods, seems to be the diet that benefits most people's microbiome. Fermented foods are interesting. Fermented foods contain live microbes. And so yogurt or kimchi, these are ways to potentially populate a gut microbiome with healthy microbes. There are some individual data showing that various microbes, whether it's in a probiotic form or a fermented food, may be advantageous to the microbiome. But generally speaking, what we're talking about here is prioritizing real food. Now, If you've heard me talk on other stages or in other conversations, I usually discuss polyphenols at length. We're not doing that today because we didn't have time, but polyphenols, as I've addressed earlier, are now recognized to exist as prebiotics. They influence the gut microbiome in a positive way. This is one of the reasons we're so fascinated with Tartary buckwheat, because it is a potent source of polyphenols. Generally speaking, though, polyphenol-rich foods, whether that's spices or chocolate, coffee, by the way, these are all ways to improve microbiome makeup because polyphenols can help to feed a healthy microbiome. What should you not do? So ultra-processed foods disrupt a healthy microbiome. What is an ultra-processed food? It is anything that you pick up in the grocery store with ingredients that you don't have really in your kitchen. These are foods that have been highly processed for shelf stability. These are foods with added sugar. So I would say we could discuss this at length. You can see my other videos on this, but generally avoiding ultra-processed food, everyone agrees, is a good idea. I would say added sugar in particular may be harmful to the microbiome. The other issue with ultra-processed foods is they're low in dietary fiber. I will just say as a quick note, artificial sweeteners, we're learning primarily from animal-based sources or animal-based research, but we don't have a consensus yet. I would say for aspartame or sucralose or these other artificial sweeteners, personally, I try to avoid those. I think we may be learning in the next few years that these are not so good for the microbiome. With better human data, there is some initial indication that that may be the case. So generally speaking, I would say avoid added sugar and avoid artificial sweeteners if your goal is to do everything you can to promote a healthy microbiome, with the caveat being we're still looking primarily at preclinical data, meaning animal data, as it relates to the impact of artificial sweeteners. Movement and the microbiome. So aerobic activity is correlated with better microbial diversity in the gut. It seems like exercise can promote a healthy gut barrier. People who are sedentary tend to have a worse microbiome, and also they tend to have more metabolic dysfunction. But like anything, we're talking about getting the right amount and not overdoing it, because excess exercise, so training for a really intense race, for example, can actually create issues with the immune system and has been linked to worse gut health. One thing that I've been talking about a lot recently is how much the quality of our air actually impacts different aspects of our health. You know, the interesting thing about the gut microbiome and our air is you wouldn't think that the air you breathe should influence your gut microbiome. However, if you think about what happens when you cough, you're actually bringing up a whole bunch of air pollutants and then you're swallowing them. So it does turn out to matter. The quality of the air you breathe does influence the microbiome. And so what can you do about this? Well, prioritizing clean air as much as possible. I've been talking about this a lot, but incense, scented candles, these are actually sources of indoor air pollution that we should be more cautious of. But just generally speaking, prioritizing good air quality is important. And then how about environment? So when we're around nature, it seems to do good things for our health. A number of different ways we can get into the specifics. We can look at data showing that children who are exposed to nature environments in schools actually have changes in their microbiome, changes in their immune system, that kids who are exposed to farms and who have more rural upbringings are at lower risk for immune-related issues, and that may relate to the microbiome. But the bottom line here is if we're in over-sanitized environments, we may be missing out on access to healthy things for our microbiome. So what do you do about it? Well, gardening, hiking, outdoor play, just spending time outside is a good thing for everyone. But I think that as it relates specifically to the microbiome, we don't want to discount the fact that we actually do get access to healthy bugs when we're spending time outside. Now, that doesn't mean going and eating some random plant you've never heard about because you think it has a good microbiome. That doesn't mean you shouldn't be washing your hands after you go to the bathroom or before meals, but it does mean that we shouldn't just be intrinsically afraid of dirt and the outdoors, because it actually may have some positive effects for our health. Now, how about substances? Alcohol's a perennially interesting topic. Moderate to heavy intake of alcohol seems to damage the gut microbiome health and promote permeability of the gut. In part, it seems to relate to certain species that influence the immune system. There is a pathway by which what happens in your gut and your microbiome influences your liver, and unsurprisingly, excessive alcohol may damage this pathway and create issues with your liver. And as a physician, I feel like it's really important for me to say alcohol isn't just something that everybody is able to completely cut off or necessarily have the ability to do that. So I think if you are struggling with this, here's my advocacy: seek professional help. This can be very helpful in terms of cutting back on alcohol or completely stopping consumption. Similarly, smoking. Smoking is associated with significant use, or significant tobacco, I should say, of any sort, is associated with alterations in the microbiome. It's associated with a number of negative effects to gut permeability, to gut immunity. Smoking is bad. I mean, I think that's a well understood thing, but specific to the microbiome, it seems to be quite caustic. And as I said around alcohol, cigarettes are addictive, and seeking professional help for pharmaceuticals or otherwise, absolutely something that you should consider if this is something that you are struggling with. Okay, let's do a recap. And for those who are listening live, I'll maybe have time for a couple of questions. But generally speaking, the main points: your microbiome plays a key role in longevity and the speed of aging. A number of recent publications are starting to unpack how various alterations in the microbiome relate to the speed of aging, and we're starting to get some signals as to what that means for what we should be doing to promote slower aging. The microbiome is far more than just what happens in the gut. It's far more than just the bacteria, and we're starting to understand how that may relate to various environmental data points that we should be feeding our bodies or not to populate a healthier microbiome. A key tool that we all need to be thinking about is that lifestyle is a significant mitigator or mediator of microbiome issues or microbiome health. And so focusing on lifestyle is the key. Looking ahead, we're starting to understand how specific subpopulations of microbes and how specific lifestyle and other interventions may help us to promote longevity by way of the microbiome. We're starting to look at tailored interventions. There are already data for specific subtypes of microbes. So, for example, we look at Lactobacillus rhamnosus CRL 1505 as an immune-supporting probiotic, but there are data for other types of bacteria in supporting other aspects of health. And we're starting to get a better sense as to how this relates specifically to immunity. There's also another category of probiotics that we didn't talk about today called psychobiotics, but it's something that I've been involved in understanding because we now understand, because of these pathways linking the gut and the brain, that the microbes in our gut may be a key way for us to mitigate mental health issues, cognitive health issues. And I think this is absolutely exploding in terms of the utility that we may be able to glean for conditions that right now our therapies are quite lacking for. So gut to brain by way of short chain fatty acids, by way of specific microbes, by way of the immune system, all are being leveraged towards the understanding of how we can target organ-specific issues. The brain, also I'll mention as before, the liver, the lungs, other parts of our body. So that was a lot of content. What I'll say to you all is, depending on when you're listening to this, our goal is to continue to produce this type of content. And so if this is enjoyable to you, please let us know. And please be sure to subscribe. We will be making more videos. I'll be doing more videos specific to slow aging. So quick teaser, we'll be talking about that in the next video around nutrition and nutrients for slow aging. But if you're watching this after the fact, also leave some comments. Let us know what you liked, what you didn't like. I'm Dr. Austin Promoter. You can follow me on my social media at Dr. Austin Promoter. You can follow Big Bold Health at Big Bold Health. And if you're interested in more content, you can come over to my channel for brain health specific stuff, and make sure you're subscribed to the Big Bold Health channel for everything immunity and beyond. So with that said, I think we may have just one moment or two to take a question. Looks like lots of questions here in the comments, but I was not able to get to them all. Let me maybe just make one specific comment related to a question that I'm seeing here. And thank you all so much for coming. It really is wonderful to have so many people care so much about science. I think the gut and just nutrition in general is a domain in which we are experiencing a major problem right now. And that problem is the confusion that comes from conflicting messages. So on the one hand, you see somebody say, we should only eat meat. Carnivore diet is the only way to go. And on the other hand, you see somebody saying it's veganism or nothing. And so what are people to do when they're hearing about these directly conflicting messages? What I think happens, and what I think is such a huge problem, is people become confused and they wind up doing what they were already doing, which is eating junk food. About a third of Americans consume fast food on a given day and not getting out of this pickle they're in where they're unsure about what to do differently. As it relates to the diets that have been best studied for overall health, I'm a huge proponent of the Mediterranean pattern diet. And some people say, oh, it's not exactly one specific diet. People in the Mediterranean eat different things. People in various places don't actually eat those foods. The reality is that the diet that is best studied for most conditions is going to be a minimally processed diet that is rich in plant-based foods. As it relates in particular to the gut, I think the consensus is clear that we don't want to be removing plant based foods as it relates to improving overall gut health. And so, my take on this would be of course, every person is different. Some people may have sensitivity to X, Y, or Z. Some people may not be able to handle fiber. Some people really need to work with a physician or another healthcare practitioner to understand their nutrient needs. But from my perspective, the goal here is not to embrace these incredibly restrictive diets at scale, and rather to say we want to embrace sustainability, we want to embrace balance, we want to embrace nutrient dense foods. So that means eating, if you can, from people who grow food near where you live. It means eating foods when you can that have been raised and harvested in a way that is maximum nutrient preservation. And it also means being honest that the goal here is not an incredibly restrictive diet that we do for 2 months and then go back to eating junk. So, just to kind of comment here on, again, some of the questions that I've been seeing, the point through all this is to try to present you with the data as I see it and as we see it, let you obviously come to your own conclusions. But I think specifically in an age of sensationalism, it's important to know that there are still some scientific consensus around what makes a healthy diet, and eating plants is part of that. So, with that said, thank you so much. This presentation will be sent out after the fact. Appreciate everybody for showing up. And Trish, thank you so much as always for putting this together. Thank you everyone for being here, and we will go ahead and shut this down. Thank you, everybody.